Hello. I decided to do my project on plant pollinator interactions, and I decided to do it on bees. When I was looking through Dryad, I actually found that there were a couple of articles regarding the effect of pollen and nectar on gut pathogen in bees. So I decided to do my project on that. So I hypothesized initially that pollen would be better at reducing the gut pathogen, better known as Strithidia bombi in bumblebees, than nectar would be. But when I started doing the calculations and by the end of this project, basically neither pollen or nectar were necessarily better at reducing the gut pathogen in bees. And that is the main takeaway from this project. And as you can see here, this is the graph that I came up with. So on the x-axis, we have the treatment type, so it's pollen and the nectar. And then on the y-axis, it's the treatment effect. So we could see that neither one was necessarily better at reducing this gut pathogen, and that's because the treatment effect was close to 1 for both of them, or it was practically 1 for both of them. And also this, I also ran a t-test, and then I got a p-value of 0.5685, and that showed that my results weren't statistically significant. And why do we care about this? So plant pollinator interactions are essential for evolution and for successful plant reproduction. Pollinators fertilize plants, and then these plants form the basis of the food chain. And then if we lose these pollinators, then that would result in flowering plant extinctions, which would affect food supply for other animals and then affect us essentially. So here's the first article that I used. The first one is the pre-infection effects of nectar secondary compounds on a bumblebee gut pathogen. So here we have the 6, so aucubin, citric acid, so on and so forth. These are all secondary compounds found in nectar. And basically to get the treatment effect, I got the average cell count for each of these after exposure to the treatment. So after exposure to aucubin or whatnot. So I got the average cell count, divided it by the average cell count of the control group. And I did that I did that 6 times. So I ended up getting 6 different treatment effects for each secondary compound. And then from there, I averaged the treatment effect, all of them together, to get one final treatment effect. And I believe it was 1.005 for nectar, which is shown here in the graph, 1.005. And then I also propagated the error. And to do that, I actually converted the total standard error into the total relative error and then into the absolute error. And that gave me the error bars that we see here on the graph for both pollen and nectar. And then for the second article, it's called Assessing Chemical Mechanisms Underlying the Effects of Sunflower Pollen on a Gut Pathogen in Bumblebees. I did the same exact thing. I got the average cell count of the gut pathogen after exposure to the treatment. So I averaged the cell count of the caprylic acid divided by the control, which was the buckwheat in this case. So I did it 5 times because there are 5 fatty acids for pollen. And then after that, I averaged those treatment effects to get one final treatment effect value for pollen, which is what we see here of 0.977. And then I did the error propagation the same way. So I converted the total standard error into the total relative error and then into the absolute error. And then by the end of this, a t-test was performed to compare the treatment effect of both pollen and nectar and to see if there was any significance. And there wasn't, because I did end up getting a p-value of 0.56. All right, that's it for my presentation. Thank you for listening, and I hope you learned something today.